Subscribe NCM Classroom for more videos. The topic is plant tissues. The topic from Class 9 Biology Chapter 2 Plant Tissues. As we know, cell is the fundamental unit of life. All the living organisms are made up of cells. It is the structural and functional unit of life because whole body is made up of cells. It is known as the fundamental unit of life because it regulates all the functions inside an organism. The cell is a Latin word for a little room. Robert Hooke in the year 1665 discovered the cell. Now let us see the types of organisms. Organisms are two types based on the number of cells. One is unicellular, where the entire body is made up of one cell, example amoeba. And multicellulars, such as plants and animals, the body is made up of many cells and different functions are carried out by different cells in the body. Now let us see the activity 1, parts of the plants and functions. Different functions carried out by plant parts. One is absorption of water from soil, exchange of gases, photosynthesis, transpiration, reproduction. Absorption of water and soil by roots and the root system. Photosynthesis is carried out by green leaves and young green pods. Exchange of gases through stomata, which are pore-like structures present on the lower side of the leaf. Transpiration is a removal of excess amount of water. It is carried out by stomata. Reproduction usually done by flowers. This is sexual reproduction. Coming to the activity 2, cells in onion peel. Items required to perform this activity: cells in onion peel. Now take a piece of an onion, now take the water and saffron in stain into watch glasses. Now carefully peel out. Now place the peel in the water. Some amount of saffron in. Now the peel is spread on the empty slide carefully. You can remove excess amount of stain by adding ethanol. Now carefully place a cover slip on it. Observe it under the microscope. In the laboratory, by using compound microscope, you can observe the cells under the microscope. All the cells are similar. Now, activity 3, cells in a leaf peel. Take a beetle leaf or a tradescantia or any succulent leaf. Tear it with a single stroke. You can easily get a peel from the leaf. Now collect the peel and place it on the empty slide. Add some amount of glycerin and 2 drops of water with the help of a soft brush. Thereby the tissue may not damage. Now place a cover slip on it carefully. Remove excess amount of water or stain with the help of blotting paper or tissue paper and observe it under the compound microscope. The cells appear like this. Activity 4, cells in root tip. Now take a conical flask or a beaker, pour some water into it up to neck, place an onion and leave it for 2 or 3 days. Now you can observe the growing roots. Now collect these roots and cut them into small pieces and place it on the empty slide. Observe it under the compound microscope. Prepare a temporary slide as we studied in the previous activity and remove excess amount of stain and water with the help of blotting paper or tissue paper. Now observe it under the compound microscope. You will see the arrangement of cells in a root tip of onion. Activity 5, growing roots. Do the same thing here. You can take 2 beakers, fill them with the water, keep these 2 onions on its surface by touching the water. Now let us see what is tissue. It is a group of cells similar in structure and performing similar functions. Usually plant has different tissues to perform different functions. Now, these tissues help in the growth repair, and other functions in plant body. Types of plant tissues: meristematic tissues, dermal tissues, ground tissues, and vascular tissues. Meristematic tissue they are small and have thin cell wall, living with a prominent nucleus and abundant cytoplasm, compactly arranged without intercellular spaces. These cells continuously divide, so they are named as meristems. These tissues made up of actively dividing cells and are called meristematic tissues. Meristematic tissues are responsible for plant growth. They are present at the tips of roots, stem, and branches. The cells present in these tissues constantly divide to produce new cells. The types of meristematic tissues. The first is apical meristem. These tissues present at the growing tip that bring about growth in length. These are called apical meristems. Depends on its location, it is named as apical meristem. The second is intercalary meristem. These intercalary meristems helps in the growth of the stem. They help the plant to grow in length. Now, the third is lateral meristems. These tissues present around the edges in a lateral manner and giving rise to growth in girth of the stem. Now let us see the activity 6. Arrangement of tissues. Usually the tissues are arranged same as in root tip and shoot tip. The meristematic tissues are usually same both in root and shoot tip areas. Because of these meristematic tissues, the plants grow in length. Now, activity 7. Preparation of temporary slide of TS of stem. This is a method of preparing temporary slide of transverse section of dicot stem. For this purpose, take young stems and take a sharp blade and make them into small and thin pieces. Care should be taken. It should be performed in the presence of your teacher. Now, collect these thin slices into watch glass that contains water. With the help of needle and brush, you can collect them. Choose the thinner section, add some amount of water to keep them wet, and use brush not to damage the tissues. Place the cover slip carefully and gently press it to avoid air bubbles under the cover slip. If we can add 1 or 2 drops of water, we can easily avoid air bubbles. And you can add safranin or toluidine blue like solutions to stain them. By staining these cross sections, we can easily observe the tissue differentiation. The excess amount of stain and water can be easily removed with the help of tissue paper. and you can use 15% ethanol to remove excess stain. Carefully remove the excess ethanol and stain. The object is very clear now, and add some amount of glycerin to keep it wet. One is stained, another slide is not stained. These two can be separately viewed under compound microscope. Under the compound microscope, we can easily observe outer layer called epidermis and the lower below the epidermis cortex, phloem, cambium, xylem, and medulla or pith, the largest part. We can easily see the structures. And as we studied, meristematic tissues present both in a root tip and a shoot tip are same. Now let us see the detailed description of transverse section of dicot stem. Epidermis is the outermost layer and has a single layer of parenchyma cells. It possesses stomata and a large number of multicellular hairs called trichomes. The second part is cortex, which is below the hypodermis. It consists of few layers of thin-walled, large, rounded or oval living parenchyma cells having intercellular spaces. Vascular bundles each contains xylem towards the center and the phloem towards the periphery, and a strip between these xylem and phloem called cambium is present. And the xylem, it lies towards the pith of the vascular bundles. It consists of tracheids, vessels, xylem parenchyma, xylem fibers. And phloem, it lies just below the parenchymatous patch of pericycle and is composed of following elements such as sieve tubes, companion cells, and the phloem parenchyma. It conducts food. Xylem conducts water, but phloem conducts food. The last part is pith or medulla. It occupies the central portion of the stem and stores food. In some dicot stems, the internal part or the medulla would be a hollow-like structure. Now let us see dermal tissues. And in the dermal tissues, the activity 8, observing dermal tissues in real leaf, we can see now. In the picture, the Rio plant is shown. And for this experiment, you can take these required materials. Now take a leaf and tear with a single stroke. You can get a peel. Now collect the peel carefully with the help of forceps. Slowly remove the peel and place it on the empty slide. Spread it and add some amount of water with the help of brush. Now you can stain or you can observe it without staining just by adding 2 drops of water and remove the excess amount of water. Observe it under the microscope. The cells are almost similar. You can observe the stomata here. These are the natural smallest pore-like structures present on the lower side. This is the structure of stomata, and you can see the 2 guard cells. and in the center the stomatal pore you can see. And now three types of dermal tissues. One is epidermis, and second one is mesodermis, and the third tissue is endodermis. You can see three types of dermal tissues. And stomata, root hairs you are observing. And coming to the ground tissues, these are three types: parenchyma, collenchyma, and sclerenchyma. Now the first is parenchyma. The chlorenchyma is another type. It is composed of living cells with a thin wall and metabolically active. Chlorenchyma is present in the mesophyll part of the leaves. These parenchyma cells have chloroplast in them, so they are green in color. These parenchyma cells carry out the function of manufacturing the food by the process of photosynthesis. The second type of parenchyma is parenchyma. These parenchymal cells are loosely packed cells with large intercellular spaces. You can observe the picture here. The larger spaces you can see. These parenchymal cells are characteristically found in aquatic plants, wherein they are involved in providing buoyancy to the plants, so they can easily float on the surface of water. The gases stored in the parenchyma can be utilized by the plant. These gases would be oxygen and carbon dioxide. Now the third type is storage tissue. This parenchyma stores water or food or waste products. Usually they store starch. The second type of ground tissue is collenchyma, composed of living cells with a thicker cell wall than parenchyma. You can see the darker regions. These are thicker cell walls. Collenchyma cells are found just beneath the epidermis and generally. And as a plant grows, these cells and the tissues they form provide flexible support for organs such as leaves and flower parts. Sclerenchymatous tissues. These cells have thick, tough secondary walls that are embedded with lignin. At maturity, most sclerenchyma cells are dead and function in structure and support. The longitudinal section and the transverse section you can see. Do you know Nehemiah Grew? He is a scientist. He worked as the secretary of Royal Society, London. He studied on internal structure of plants in 1664. He gave the term parenchyma for the pithy part called medulla. He initiated the study of tissues. The study of tissues is called histology. He wrote a book, Anatomy of Plants, in the year 1682. This is about Nehemiah Grew. Now, let us see vascular tissues. Vascular tissues are composed of more than one cell type, are generically referred to as complex tissues. They help in the transportation of material, so they are also called transportation tissue. Vascular tissues are usually two types. One is xylem, another one is phloem. Xylem is an important plant tissue as it is part of the plumbing of a plant. That means transportation of water. It carries water and dissolved substances throughout and consists of a combination of parenchyma cells, fibers, vessels, tracheids and ray cells. Coming to the phloem, it carries dissolved food substances throughout the plant. This conduction system is composed of sieve tube member and companion cells that are without secondary ones. As we studied about the xylem, the phloem also the same function it carry out as a plumbing part of the plant. Vascular tissues carry water to the height of nearly 200 feet in eucalyptus plant. Vascular tissues also carry water to the height of nearly up to 330 feet height in redwood trees. Subscribe to our channel NCM Classroom for more videos.